Yeah. Hello. So welcome back, everyone. This is the final session for this season, and our first speaker is Barnabe Ledoux. He's he's PhD student of David Lacoste and from ESPCI, and he'll tell us about compositional memory matters for early molecular systems. Please.
Yeah, thanks a lot. Thank you all for being here. It's really an honor to be able to present our work today. And I want to thank the organizers for inviting me. So today I will talk to you about this work that we published recently in PNAS. So if you want to have a look afterwards, now it's published. So we did it together with my supervisor David Lacoste and with Ryo Mizuuchi, Ryuka Kuwabara, and Norikazu Ichihashi in Japan, who performed the whole experimental part. So there are many very interesting experiments in this work. Today we'll mostly focus on the theory because it's the part that I worked on, but I will try to give you also insights on the experiments. Um, I want to start with a bit of context quickly. So, this work has been inspired by a whole literature that tries to find the constraints and the requirements to have the emergence of life. So obviously there is a whole literature on that. And in particular, we were interested in understanding how we can avoid what's called an error catastrophe. So this is mostly due to the work of Eigen, um, where he basically he finds a condition, this inequality here, under which you can have the coexistence of parasites and hosts without having parasites taking over the system. But this is the so-called eigenparadox. But because like if you have too long molecules, so if L is too large, you expect to have an error catastrophe. So meaning that basically the parasites start taking over your system. And so he found this inequality as a constraint and that you need if you want to have long functional molecules that survive, basically. So the dynamics that he considered were a bit different from the ones that we will consider here. So it's not really the same parasites and the same hosts, but we are interested in the same issue, that is, how do you prevent your system from being invaded by parasites? And it's also related to many, maybe a bit more recent works. So, for instance, there is this work by Philippe Engue. So it's really recent, as you can see, 2025, where he details a scenario for, like, a possible scenario for the emergence of life, and he basically details different steps and different requirements, and he focuses on the importance of compartments, of having at some point your molecules that are close by spatially. So it can be because of diffusion limited phenomenon, for instance. If you are in a porous material or in a viscous material, you can have limited diffusion, putting your species close by, but it can also be the result of a more complex membrane, for instance, or a closed droplet, where really you encapsulate some individuals together, and they can't interact with what's outside the membrane. So it can be through lipid-lipid phase separations, or many other phenomenon. In the work, we in the theory, we don't really we don't specify a specific membrane. We just need to have a closed compartment. So this will be an assumption in the theory, as you'll see. And there is also this very important work so done by Ryu Mizuchi and collaborators, where they study the evolution of a network of hosts and parasites. So you see that they start with one host, HL0, and they do what's called they do different steps of compartmentalization. I will detail afterwards. But with these compartments, they are able to see the appearance of a complex network of different species, and they all coexist. So you don't have parasites taking over the system. You don't have uh, parasites disappearing either. They really coexist with the host. And you see that the networks seem to be more and more complex. So we wanted to understand that with a simple model. So before moving to the model itself, I just want to give you an intuition, because maybe it's obvious for everyone, but quickly I want to give you an intuition why compartments are important and why they can prevent parasites from taking over the system. So here we will consider replicases or hosts, but I will call them replicases from now on. So they are molecules of RNA of about 2,000 nucleotides that are able, through a mechanism that I will mention after, they are able to self-replicate, basically, to make more of themselves. And so they are functional. They are quite long, as you can see, I mean, compared to the parasites. And so those are the replicases. And you also have parasites that are shorter molecules, about 500 nucleotides long. And those they can't replicate by themselves, but they can be replicated by the replicase. So that's the key assumptions that you need to keep in mind. And so now if you just put them together, what do you expect? Basically, the parasites are shorter, so they can be replicated much faster by the replicases. And so if you assume that all species can interact, you will get the pool will be filled with parasites quite quickly. And you won't have time to replicate many replicases. Maybe you can hope to have a few, but if there is dilution, for instance, at some point you will lose all your functional molecules because they are much slower. Now, what happens if you have compartments? So assume you have droplets or you have membranes or anything that can be closed and encapsulate only a few individuals. So a few molecules, like one or two molecules. Then you can have different things happening. You can have parasites that are isolated. But the parasites, they need the replicase to be replicated. So if the parasite is isolated, it just won't do anything, and it will remain alone. Now you can have a replicase and a parasite together. So in this case, the parasite will fill the compartment, but it will only fill the compartment, not the whole system. So it remains localized in a way. And you can also have isolated replicases, and in this case, they can replicate freely because there is no parasite to take their resources, basically. So this is the idea, and you see that now with compartments, you really favor the replicases. So it's a bit sketchy. Now let's try to put it in equations. So I will try to detail the different assumptions that we have. First, in this work, we wanted also to model mutations because we wanted to be able to understand how you can, from only one or two species, go to a network of more and more species, and how you can have all of them coexisting. So to do that, we had to model mutations at some point. And to do it properly, we used the same work by Ryo Mizuuchi and collaborators from 2022, um, where they built a phylogenetic tree of their different species. And you see that you can have what we call model A. That is that you can have replicases, so HL0 and HL1 are replicases. You can have a replicase which mutates to give another replicase. So it's still a long functional molecule able to self-replicate, but it's a different molecule, and so it's a different replicase. But it's still a replicase, and you can also have parasites that mutate to give different parasites. So what is the difference between these species? It's mostly that, for instance, PL2 will be able to use mostly HL0 to replicate, but not much HL1. And PL3 will be able to use HL1, but not HL0, for instance. So you have this kind of specificity between parasites and replicases, and that's what makes the species different. So this is model A. Replicase mutates to give different replicase, and parasites mutate to give different parasites. And then we need also assumptions on the maturation step. That is how the compartment itself evolve depending on its initial composition. And you see here that we have also different assumptions. So I will be a bit quick on that, but we have efficient mutations. So meaning that we assume that mutations gives molecules that have higher replication rates. Basically, it's an assumption that we can that is not necessarily here in the experiments, but we can switch the role of mutants and wild type, and so in the end, it's just to simplify the dynamics in the theory. Some parasites also have a strict specificity, so that's what I was mentioning just before, meaning that some parasites will only be able to use a specific replicase to replicate. And we have also the assumption of aggressivity of parasites, meaning that they replicate much faster than the replicases. Okay, then we have model B. Model B is also observed in the phylogenetic tree. It's when you have a replicase that mutates to give a parasite. So for instance, through deletion, recombination, you can have a large part of the molecules which is deleted, and you end up with shorter molecules that are parasites. And so we also model this, and we keep the same assumptions and the dynamics. And we did this because we wanted to know if there is a strong difference between those two models. In the end, theoretically, you get the same kind of behaviors with model A or model B. But yeah, we studied both. So now we model this cycle. So basically, this is the first assumption on what's done experimentally, and later I will detail how we improved it. But so let's start with this. Basically, we assume that we start with many compartments. So let's start on the left of the sketch. You have many compartments with a few molecules inside, 1, 2, 3, but not more. And the distribution of the contents is random. And then you let all the compartments maturate. So depending on their initial composition, they will end up with a final composition. And then you have what's called pooling. So we assume that we can put all the individuals together in a common pool. So we lose basically the memory of what was in a specific compartment. We just put everyone together. And then we have dilution, so we remove randomly some molecules. And from this, you are able to start again through compartmentalizing your system randomly again. So as you'll see, there is something that is different in the experiments, but this is the first assumption that we considered. And you can put all of these steps in equations. So compartmentalization, as I said, is just a random Poisson binomial process. So you just fill your compartments according to a Poisson binomial distribution with the different species. Then you have maturation. So maturation, it's also in principle a stochastic step because the reaction rates are stochastic, and you can model this with a Gillespie simulation, for instance. But in principle, we use a deterministic assumption for this step. I won't go into into the details now, but yeah, it helps simplifying the process, and we show that basically it's equivalent to the Gillespie simulations. Then we have pooling. So pooling basically it amounts to just put everyone together. So forget about the individual composition of the compartments and just compute the average fraction of each species in the whole system. And dilution, you just divide the total number of molecules by a given dilution factor. And then you're ready to start again. So we modeled these different sets of equations, and we obtain this kind of dynamics. So you see, we get cycles. So I'll try to explain quickly why do we get cycles. So first, X is the fraction of replicases in your whole system. So it's just the number of molecules that are replicases to the total number of molecules in your system. So if X is 1, it means that you only have replicases. If X is 0, you only have parasites. And lambda, it's the average number of molecules per compartment. So if lambda is 1, you have in average one molecule per compartment. And it's the case on the sketch on the right. You in average, you have one molecule per compartment. So why do we have cycles? Let's start at the bottom of the cycle. So assume you have a low value of lambda, close to 1 or even below 1. Then it means that you often have isolated molecules. And if you have isolated parasites, they can't replicate. But if you have isolated replicases, they can replicate. So this situation is very, it favors strongly the replicases. So that's why X is increasing in this part of the plane. But then if you increase X, you will start having many replicases in your system, and you will increase the population because you will have more and more replicases, and so more and more compartments will be able to grow, basically. And so the population will increase, meaning that lambda will also increase because in average, you will have more molecules per compartment. But if lambda increases, now you will have, as you can see, in average, 2 or more molecules per compartment. And so you will often have one replicase and one parasite. And in this case, the parasites, they can replicate. And so this will favor the parasites. So you will go towards lower values of X because you lose replicases. And if you lose too many replicases, at some point your system can't replicate anymore because you will have many compartments without replicases. And so most of the population will die out, will be diluted out rather. and so you will decrease lambda. So yeah, that's for the explanation of the cycles. I hope it's a bit intuitive and a bit clear. But you see, then depending on your initial state, you do cycles, and at some point you seem to reach a limit cycle, which is in red on the two graphs. So on the left, it's rather a steady state. It's really a single point. On the right, it's a limit cycle. And we obtain different limit cycles depending on the conditions. So here on the two upper panels, they are for two species. So you only have one parasite and one replicase. And you see that in this case, we get clean cycles. And the two columns, they correspond to different values of the ratio of the carrying capacity to the dilution. So if you have a larger carrying capacity, you make larger cycles, basically. And you see that in terms of dynamics, it corresponds to oscillations between parasites and replicases. And on the bottom, the two bottom panels, you have four species now. So you have two parasites, two replicases. And you see that the cycles get more complex, but we still have cycles. And in the end, we also get oscillations in the dynamics. But one thing we can notice is that it seems that for the same ratio of carrying capacity to dilution, the case with four species reaches a steady state more easily. So it seems that the more species you have, the more your system is stable, basically. Okay, so that's the kind of dynamics we get. Now I will just tell you a bit about the experiments. So the way it was done experimentally by our collaborators in Japan is to use RNAs that can be translated. So HL1, HL2, they can be translated to form polymerases. And then those polymerases, they can perform the replication. And so they can replicate the different parasites or the replicases itself. So basically, you need to go through a step of translation. But the theory is quite general. We don't the theory does not rely on this translation step, but it's required for the experiments because yeah, it's the way we can get this kind of self-replicating molecules, but in a way they are not completely self-replicating because they need to go through this step of translation. But still, so we have those great experiments that we can compare to the theory, and you see that we managed to reproduce quite well the kind of oscillations. At least we really capture the period of the oscillations, and we really capture the fact that those four species that are a priori aggressive to each other, they can coexist in this system by oscillating like that. So yeah, so that was a first cool result. But then we wanted to understand a bit more what happens experimentally, and in particular, what's done experimentally is closer to this. So you have the first maturation or replication step. It's the same as before. But then it's a bit different because dilution is done by removing some droplets randomly and by putting new empty droplets in the system. So now you have a population of full droplets and some empty droplets all together. And then you perform a stirring step where you mix the contents of your different molecules, of your different droplets, sorry. And this mixing step or stirring step, actually, it's quite important, and it corresponds to the pooling of the previous model, only in the limit where the stirring is very strong. Because you have two limits. Either you have a weak stirring, so you don't mix at all the molecules. In this case, you will keep your empty molecules, your empty droplets, sorry. So if you don't mix your droplets, you will keep your empty droplets empty, and the contents of the different droplets will not be modified. Now, if you do a very strong stirring, so you really homogenize all your droplets, then you lose the memory of the compartments because you mix everything. And so this corresponds to the former limit. S goes to 1, it corresponds to the perfect pooling. But now we were interested in intermediate value of this parameter S to see what's the effect of this compositional memory. So to give you an intuition, if it's not clear yet, so assume that you don't mix your droplets, you will start with a very heterogeneous composition of droplets, like that. So some are empty, some are full. Now, in this case, you tend to favor parasites because in the empty droplets, nothing happens. And in the other droplets, you often have more than one molecule. So it means that the parasites will more easily find a replicase. So this situation is the best for parasites. Now, if you have a very homogeneous distribution, and a low enough number of molecules per droplet, you will recover the case where you often have isolated parasites. And so in this case, as we've seen before, you favor the replicases. So it seems that increasing the stirring is the best way to help replicases surviving. So we can do the same kind of cycles, putting everything in equations. So the equations for maturation, they remain the same. The equation for dilution, it's also the same, basically. But in practice, what you do is you remove some compartments and you replace them with empty compartments. But mathematically, it corresponds to just reducing the average number of molecules per compartment. But what's really different is the stirring step. So now you need to keep the trajectory of each individual compartment because you have a compositional memory of what was the composition of your compartment before the stirring step. And what you see is that, so here in dotted lines you have the limit S equals to 1, basically. So the perfect pooling limit. And in solid lines, you have the dynamics with an intermediate value of S. So a given level of compositional memory. And you see that it really seems to favor parasites in the end. And if you go to S equals 1, you recover the perfect pooling limit. And now what we did to understand the mechanism is that we so in full lines you have the average over whole compartments, over all the compartments, sorry. But now in purple dots you see the compositions of each individual compartment, and you see that when S is intermediate, you really have an heterogeneous distribution. And when S is close to 1, you go to a very homogeneous distribution. Either you are empty or you're full, basically. That's the idea. Okay. So to test this experimentally, so our collaborators in Japan performed designed this experiment with fluorophores. So I won't go into the details because I don't have much time left. But basically, you have green fluorophores and red fluorophores, and you can quantify the amount of stirring through a renormalization process. The calculations are not so easy, but in the end, you can quantify the level of stirring by looking at the distribution of colors. So if you have two peaks, one peak for the red and one peak for the green, it's that you don't have a strong mixing. But if you start mixing a lot your compartments, you will get orange or yellow. And yeah, so just by looking at the distribution of the colors, we can get a value of S. And so that's what we did. And this way we were able to associate with the value of the parameter S a value of the steering strength that is done experimentally in kilo rotation per minute. Uh, so that's what we did. So again, it's a bit complicated, but the point is that it allowed us to compare experiments to theory by associating a value of the theoretical parameter S to an actual value of the rotation per minute that are done in the lab. And so this way we were able to compare what's expected for the fraction of replicases X as a function of the steering S. We compare experiments and theory, and you see that we get the trends, and we get even a quantitative agreement within a given error. But it seems that we really see that increasing the steering favors your replicases, and it was observed also experimentally. So this is, if you have a small steering, you favor the parasites. If you have a strong steering, then you favor the replicators, as I've told you before. So now I will conclude. So as you see, there are many things in this work. But mainly the key message is that compartments along with steering seems to be a way to sustain replicases in your system. So compartments alone, if you don't homogenize your population at some point, it's not enough in a way. You need this steering step to keep replicases in your system. And we also observed other conclusions, like the fact that it seems that more species tend to stabilize the system. So it's something that maybe we would like to explore a bit more. And so now I just want to thank David Lacoste, Ryo Mizuuchi, Ryoka Kuwabara, and Norikazu Ichihashi, and thank you for your attention.